So good morning, everyone, and welcome to the Elixir Czech Republic Friday Coffee, which is a series of seminars and short lectures about the tools and other services. This time we have a honor to welcome Marian Novotný from Charles University, and he will talk about the PrankWeb, which I suppose is something designed for the ligand designed to the proteins. So Marian, please, the floor is yours.
Thank you, Jurka. Thank you for coming this Friday. I will be presenting a tool which I have to say is mostly work of someone else, so it will be a bit unusual. But so that's why I start with acknowledgements. Okay, it works now. So this work was mainly conceived and done at Mathematical Physical faculty, and it went through the hands and brain of David Hoxha and Rado Krivák, and later Lukáši and Dele. And then we formed a joint bioinformatics group between Faculty of Science and Mathematical Physical faculty. So we joined the project. That's where I come in, but there are other people who participate on this. It's Petr Škoda, who is maintaining the web server I will be talking about in a while, and David Jakubec, who you heard already in this Friday Coffee seminar series. And he's taking care of the conservation pipeline I also will be talking about. So what was the motivation to do the PrankWeb? There were many, honestly, but one of them was that nowadays the 3D structures can be solved and are solved even for proteins which we know very little about. This previously was not very typical because it's extremely complicated, extremely expensive. So the 3D structures were solved only when you knew why you were doing it. But now it's possible to do it also for the protein of unknown function. And indeed, there are quite a few already in the protein data bank. There are over 2,000 structures which are deposited with the name, including word putative or possible or hypothetical protein. So we obviously need new tools to describe function or evolution of these proteins using 3D structure information. So, and that's happening. There are a lot of new tools. And one of the tools you could have is the one which would predict ligand binding sites on the protein structure. And you can use it for proteins of unknown function, but you can also use it in the drug design or protein engineering. That's the way we went. We tried to develop a tool for prediction of ligand binding sites on the protein structure. And that's our tool. It has two names to be more complicated. So one of them is P2Rank. That's the original algorithm developed by David and Rado. And later we made also the web version, which is called PrankWeb. So it's a method to identify surface regions which are capable of binding unspecified small molecules. So we do not predict which ligands is going to bind in the position, but we say this position is likely to bind something. What we need as an input is a protein structure, and it can be modeled protein structure, but it has to be 3D coordinates. And the output is a list of surface regions which are likely to bind some ligand. And I will walk you through the whole thing. Yeah, and how we do it. It's a machine learning method. It uses random forest algorithm to discriminate between the binding and non-binding sites. So how do we start? We started by building a model, and for that you need known protein ligand complexes. We started with a relatively small data set called CHEN. It had about 200 structures with the ligand, containing about 450 ligands. I think they were quite carefully selected. Each scope family, so that's a classification system for the structures, was represented by just one structure, and we tried to avoid all the ligands which are not biologically relevant, like salts, solvents, and so on. And for this data set, and each structure in the data set, we made a sort of a net, which we used to cover the structure of the surface of the structure. And the net was described by mesh of points. And for these points, we can calculate a vector of physical chemical properties and structural features. And for each point, you could say whether it's binding and non-binding based on the protein ligand complexes. And when you do that, you can build a model which for a given vector will say what is the probability that this part of the pocket is binding. So what features do we take? We take a physicochemical properties like hydrophobicity, charge, and so on, but we also take a geometrical properties, and indeed this one seems to be the most important one. One of them is called protrusion. It basically measures how many atoms are within certain distance from a given point in a mesh, and altogether there are 35. And then we go into detection. So you take the new structure where you are interested in where could the binding sites be. You cover it with the mesh again and apply the model. And for each point in the mesh, you can calculate the probability. We call it ligandability score. And then we filter out points with low ligandability score and cluster the remaining points, because our method is basically not a residue based, but a pocket based. So we tried to cluster the points, so we create some pockets, and then we score the pockets. Just accumulate the ligandability score, get some raw score, and then you can go back, and from these points you can map them on the residues. So in the end, you will get an information about which residues are part of the pockets. So that's the in short the algorithm. And then for the PrankWeb, we introduced another step where we gave another parameter next to the geometrical and physical chemical, and that's a conservation feature. And this conservation feature can be used either as another parameter in the model, or it could be a post-processing step where you could rescore the found pockets and prioritize based on the conservation. So we either, that's quite a complicated chart, but we either try to find we calculate the conservation from multiple sequence alignments, and either we can find it in HSSP database, or we calculate it ourselves using PSEBLAST in SwissProt or in UniProt database. And then we calculate the conservation. This step is actually quite slow, but it helps to improve the results. The idea is that the more conserved sites are typically the ones which are more likely to bind ligands. So, okay, so how does this algorithm perform? There are two standard sets of ligand structure complexes which are used, which is very useful because then you can compare different methods. And this one, coach, is quite a small subset of about 400 structures of monomeric proteins, while this whole of 4K is much bigger, 10 times bigger, and it contains also multimeric proteins. And the measure we use is not the typical one, sorry, and not the typical one. based on the residue level, but as we do the pocket-based prediction, we use different one, which we call the success rate. And in principle, it's if you take a center of the pocket, how many atoms you will get, will you get the ligand within the 4 angstrom threshold from this center of the pocket. And you can see that P rank, P2 rank is actually, from those we tested, is doing the best. And that if you use the conservation as a parameter, it gets even a bit better. Not in this case, but in all 3 other cases, it does. So we also show that the conservation can improve the performance. But it's also important for one more thing, and I will show this to you when we get to actual program. Okay, so you can get it either through GitHub, where the full thing is available, or it's a web server I will show you in a while. And the other thing where you can get it is the PDBE knowledge base. It is the thing you heard about during the Friday coffees. It's an attempt to look at the protein structures through the protein lenses. So you can find all the structures for a given protein. You type in UniProt ID and you get all sorts of information about structures for a given protein. And it integrates a lot of different tools, and we are happy that it also integrates our tool, which is P2 rank. And here you can see that's the output for human actin. And here you can see from the crystal structures the ligands which are bound, and on this line you can see, so that's a residue level mapping, and you can see that it matches actually quite well with what is known. If this is not interactive, but if you would go to the web page, you would see which pocket this is. So on this graph, it's not color coded, but you can get the information for which pocket each residue participates. And we have a lot of plans with this thing. So it has not been trained for prediction of metal ions, for peptide binding or nucleic acid binding. And sorry. And we would like to train it for metal ion binding and nucleic acid binding. We built new data sets, and we are in the process of training the model. So this should come. And we also want to train it as a tool for identification of cryptic binding sites. Cryptic binding sites are the sites which change conformation substantially between the apostructure and the holostructure, between structure which is not binding ligand and the one which is binding ligands. And these are tough nuts to crack most of the time. So we would like to train for this. And our current web server is actually quite slow when we try to calculate the conservation. So we are replacing the conservation pipeline to make it much faster and also a bit more precise. So that's basically it from this part of presentation. And now I would like to show you how it works very briefly. It's a very simple thing. So you either have the structure you want to predict, so you can upload your own structure, or you do the one which is in the PDB, and you can do it for a single chain, or you can do it for the whole structure. If I remember the structure I want to show you, yes. So if you type the code, it will tell you how many chains it has, and then you can restrict for individual chain. And there is another option you have. You can either run the conservation or you can skip it. You would skip it sometimes because it can be really slow if the structure has not been pre-calculated. And but you can also help it by providing your multiple sequence alignment. Sometimes you think you have really good multiple sequence alignment, and most of the time it will be better than the one we can produce automatically. So I will just run it now without the multiple sequence alignment, without the conservation. And one of the big advantages of the algorithm is that it's really fast. So you see that it provides the results almost instantaneously. So what you see here now is the structure of KRAS protein. And I chose this example because it's quite relevant for precision medicine, which is another area where ELIXIR Czech Republic would like to be much more active. And KRAS is one of the oncogene which is the most mutated in many cancers. And there is one particular mutation, which is glycine to cysteine on the position 12. And this one appears in almost 10% of lung adenocarcinomas. It's by far the most common mutation. And for a long time, it was thought that it cannot be used as a drug target, but some clever people came with an idea that the cysteine, which is the mutated residue, could be used for covalent modification. So they indeed developed a number of drugs which are now tested for clinical use. And one of them is from Amgen. It's called AMG 510, and that's what you see here. But the output from us is that you get 4 pockets. In this case. Each of them, they are ranked by the score, and there is just some, it can be described by absolute number or some probability. And if you calculate conservation, you would get another score for the conservation. And you see that the first pocket is not the one with the ligand, but important thing, we use mole star to visualize the structure. So it's another beautiful, I think, cooperation between different groups in ELIXIR Czech Republic. And mole star in this case is doing funny thing. We will have to report to David Sehnal. So one of the ligands, which is here, you can see it's a GDP. That's the main ligand for KRAS because it's a small GDPase. Is not shown as a ligand, and I don't know why, but the ligand binding sites around is quite clear. And already the second pocket is the one for this ligand. So in this case, you can see how well this is predicting the known binding sites. And I will show you what happens when you run it with the conservation score switched on. The output is very much the same, except there is it's annoying part of, except there is just three pockets. So one of the lousy one went away, and that's another big advantage which we see very often. If you use the conservation, you filter out a lot of very weak predictions for the ligands. So that's pretty much it. What you can do here, and it's also important, if you don't like this visualization, you can download all the results and you will get the things colored for PyMol. So if you do download here, you will get it. And here you have mapping with Proteal for the sequence as well. And with that, I think that's all I wanted to say. So thank you very much for your attention.
Marian, thank you. Thank you for your presentation. It's a very interesting tool. I got 2 questions. So first is, what if the unknown protein has no binding site? Is the Prank tool able to distinguish the binders from non-binders?
I haven't tested this very strictly, but the score, the pocket score, should be indicative. It's not. it's not perfect in a sense that the big pockets will get much bigger score than the small pockets. But if you get something, well, first, it's very unlikely that some protein structure will not bind anything.
But on the other hand, you can have also the protein-protein interaction, but I
Yes.
don't know if you are able to distinguish between the ligand binding and the protein binding.
Well, we don't know. We haven't trained it for recognized protein-protein interactions, and from what I saw, it did mostly predict the ligand binding sites, but we will have to investigate this more in detail. But what I wanted to say, the pocket score can be often really low. So if you would get the best pocket score with value like 1 or 2 or 0.96, that it's very unlikely that it's actual pocket.
Okay. And I think there is quite a logical question. So if there is a fingerprint of either the binding site or binders, are you able to identify the binding site using these fingerprints? Because the most chemical database is providing the fingerprints of the ligands. So if you know it, and if you would know also the fingerprint of the protein binding site, are you able to somehow match it?
We haven't trained it for this. I think it should be possible if we have enough data. So I think for certain ligands or type of ligands, we should be able to do it. But at the moment, it's just very general prediction tool. So it has not been trained for any specific ones.
Okay, so yeah, there is no other question. So Marian, thank you again very much for the presentation and for all of you who actually was presented at the Friday's Coffee. So we now have a vacation. So we see each other on September. So have a nice vacation and have a good day. Bye.
Thank you. Bye.